Okay. Hey guys, I guess. It's Jeff. So, I'm going to make the video for big idea number 3. And I'm going to look at Enduring Understanding 3D, which is mainly about communication of cells. So, I'm going to go over cell communication, signal transduction pathways, and evolutionary significance of cell communication. And I basically ripped off 3 Bozeman videos and just shoved them into one video to summarize them all, and basically go over the main points. So basically, I'm going to go first over cell communication. So there are 3 types of cell communication. There's the no distance, short distance, and long distance. So for no distance, they're basically cell to cell contact, direct contact, and basically it uses antigen presenting cells, antigen presenting cells, or basically one cell. A cell sends a message to another cell. Yay. An example of this are plasmodesmata that plant cells have, that they used to send signals directly from one cell to the other cell. Yeah. The next one is short distance, and they're basically local regulator, like neurotransmitters. And this neurotransmitter are used when it's basically one neuron to neuron by synapse. And if you remember, it was like a bunch of pictures where it was like a neuron and neurotransmitters, and then another neuron with like receptors and stuff. So the third one will be the long distance. The example I used was hormones, like growth hormones. Where it goes from the brain, and it go to like the liver or bones or other body cells, and etc. So after the cell communication is the signal transduction pathway. So signal transduction pathways. And there's basically a general flow to this. So it first goes from the messenger to the receptor, to transduction, to the secondary messenger, to the target cells, or like what it's trying to make the cell do, basically. Not the actual cell targets, because it's inside the cell already. So basically for this idea, or enduring understanding, whatever we'll call it, I'm going to use epinephrine, how it affects liver cells. So epinephrine to liver cells, and this is going to be mainly just drawn more than written out. So first there's this epinephrine, right? And here's like the plasma membrane. Keep it really simple. And there's this G protein, whatever you want to call it, whatever. And this epinephrine binds epinephrine to this G protein receptor. So when it binds, it causes G protein receptor to change, like it changes. And the G protein receptor has these alpha subunits. And when the G protein changes, the alpha subunit is released and then goes to a different protein. And in this case, the different protein is the adenyl cyclase. So let's draw a little adenyl cyclase, whatever you want to look at it. Adenyl cyclase. So this adenyl cyclase. So the subunit activates this protein, and it's an enzyme, and this enzyme so this enzyme converts ATP, I'm hoping I'm getting this right, into cyclic AMP by losing two phosphates, hence the mono. And this is a secondary messenger. So this camp, it targets a protein kinase, and let's just say this is a protein kinase. It targets it. Protein kinase. It targets the protein kinase to create a cascade of energy, and basically the protein kinase gets activated when it becomes phosphorylated from ATP. So like here's another ATP, yay, and it phosphorylates. So yeah, and then this protein kinase moves over to this thing called, it's an enzyme called, it goes to this phosphorylate, and it activates it by dropping off a phosphate. And what it does is it breaks down glycogen. So let's just draw some glycogen. Oops. Glycogen. The phosphorylate breaks down the glycogen and causes it to go out of the membrane as glucose. So yeah, that's basically a signal transduction pathway. I actually don't know if I said everything right. I just try to do what I remember to watch the Vosen video. But then the last one is the evolutionary significance of cell communication. Evolutionary significance of cell communication. So this one, I'm going to compare single-celled organisms to multicellular. And I'm going to use examples like a single-celled organism will be a bacteria. So a single-celled organism is they respond to the environment, like the quorum sensing, which is basically where one bacteria can sense like if it's like surrounded by other bacteria, and like how many bacteria is surrounded by and stuff. So basically how this works is one bacteria little bacteria, gives these things off called an autoinducer. It's a protein. Autoinducer. So yeah, but then when there are several bacteria, they have all these autoinducers, all these autoinducers. This bacteria will pick up the other autoinducers that are from the other bacteria, and it will cause a signal transduction pathway, which back to this stuff, except it's not epinephrine, it's the autoinducer. But it causes a signal transduction pathway to transcribe a gene. So that's basically the general idea for this one. So for example, a glowing bacteria, the one that like glows, like bacteria, it's like, oh, glow, yay. They glow in a colony because they won't glow by itself, but when they're in a colony, they'll receive these autoinducers from the other bacteria, which transcribes a gene for it, causes signal transduction pathway, and transcribes this gene to create